I thank you. I wanted to welcome all of you to the CNS infection session. And we have a lineup of some really terrific talks. The first one on viral diagnostics, then we have one on emerging infections, and then on effects of viral infections on cognitive function. And then we have two posters that we've selected that will be presented. And so the first talk is by Dr. Michael Wilson. And he is the Debbie and Andy Ratchleff Distinguished Professor in Neurology at University of California, San Francisco Institute of Neurosciences. His lab studies the ways of applying new methods for diagnosis of viral infections. And he uses metagenomic and immune repertoire sequencing techniques, as well as phage display antibody discovery techniques to understand the causes and immunopathogenesis of autoimmune and infectious causes of meningoencephalitis. He has received multiple awards during his career, and including the Trailblazer Prize from the Foundation of National Institutes of Health, which is an extremely prestigious prize for young investigators. And with that, I'll hand it over to Dr. Wilson.
Hello, my name is Michael Wilson, and I'll be speaking about viral discovery in CNS inflammatory diseases today. These are my disclosures. Encephalitis, or acute brain inflammation, accounts for about 20,000 cases a year in the US, with a similar number of patients suffering from meningitis. Although not common, these patients are disproportionately burdensome to the healthcare system, with about $2 billion in inpatient costs each year, 10% mortality acutely, with additional mortality in the delayed weeks and months afterward. And many of the survivors have significant long-term disability, including significant cognitive deficits, as well as motoric and sensory and coordination deficits. One of the biggest challenges of managing patients with encephalitis is frequently not knowing what the underlying disease is. So depending on the study, both in the developing world and in the developed world, about 50% of patients have an unknown cause to their encephalitis, with the two most common categories of causes being infectious and autoimmune. And not knowing whether patients should be immune suppressed for an autoimmune disease or treated with antimicrobials for a possible infection can really paralyze and hinder getting them effective treatment in a timely manner. One of the reasons and challenges for diagnosis in encephalitis, especially infectious causes, is the growing number of emerging and re-emerging infections that can cause neuroinvasive disease. And this is an incomplete list here. And of course, this is all on our minds the last couple of years with SARS coronavirus 2. So the typical approach to diagnosis involves the formulation of a differential diagnosis based on a patient's symptoms, their imaging findings, and the physician's knowledge base of possible infections, and then a series of candidate-specific or pathogen-specific tests are sent off to rule in or rule out the hypotheses that a physician makes about a particular case. Those hypotheses and those types of tests can't anticipate an emerging infection that for which a diagnostic test may not yet exist. This was really vividly highlighted with the Zika virus outbreak in Latin America a few years ago. It's now estimated that Zika virus had been circulating in Brazil for a full 18 months before it was detected, and nearly 2 years before it was recognized as a cause of the spiking cases of microcephaly and Guillain-Barré syndrome in Brazil. And again, this highlights that because Zika virus had not been seen before in Latin America, there weren't pathogen-specific tests available in that part of the world. And even if they were available, it wouldn't have made sense to send them because it wasn't known to be circulating in that region. And that's why we and many others have worked on the development of metagenomic next generation sequencing as a tool to detect viruses and other neurologic infections. And this tool has the advantage of being able to survey in a rapid and cheap manner all the nucleic acid that's present in a sample. In our case, the most common sample is cerebrospinal fluid and occasionally brain tissue itself. The advantages of metagenomic sequencing are that it can identify novel or highly divergent pathogens like SARS coronavirus 2, pathogens that haven't previously been linked with a clinical phenotype, so a diarrheal illness that we're now recognizing might be causing a neurologic disease, rare pathogens that are known to cause encephalitis, but that aren't on the differential in a particular case, or potentially as a test to rule out an infection in suspected cases of autoimmune encephalitis. So this is a case of a 63-year-old man from Cape Cod who had lymphoma and was treated with rituximab, a B-cell depleting drug. And he presented with a week of fever and testicular pain, was given antibiotics for possible bacterial infection and sent home, but then came back 3 days later with neurologic symptoms as well, including slurred speech and gait imbalance. He also had a stiff neck, and he got a spinal tap, which showed that he had inflammation in the spinal fluid and an increased number of white blood cells and an elevated protein level, suggesting that there might be an infection present. His MRIs were quite abnormal. You can see here that there's edema or swelling in the cerebellum that's impaired CSF outflow, and that's causing what's called hydrocephalus in the lateral ventricles and flow of spinal fluid because of that high pressure out of the ventricles into the brain parenchyma. In addition to that swelling, the patient also had what's called contrast enhancement, meaning evidence of contrast leaking into the meninges around the cerebellum, again, supporting the idea that he had a meningitis as well as an encephalitis, so a meningoencephalitis, and primarily involving the cerebellum. So deep sequencing in this case identified Powassan virus, which is a tick-borne flavivirus that can cause life-threatening neuroinvasive disease. This particular case was a fatal case. And it's been recognized for over 60 years, but is increasingly recognized as a cause of encephalitis in the northeastern United States. And in this case, the patient was B-cell depleted, so he had a delayed antibody response, and so the Powassan virus antibody test was initially negative, but direct detection with deep sequencing was able to diagnose this neuroinvasive flavivirus infection. In addition to just detecting the virus, as we've seen extensively with SARS-CoV-2, you can recover enough of the genome frequently to do phylogenetic analyses to understand more in a more high resolution way exactly what lineage of a virus a patient has and from whom they may have contracted it. And this is another case of a patient with a more chronic encephalitis, which is a less common manifestation of encephalitis, but can be equally severe. So this is a patient of mine, a 27-year-old man who developed a spinal cord inflammatory syndrome a few years prior, thought to be autoimmune, that left him paraplegic. And this was his original spinal cord imaging. You can see the extensive scarring throughout the cervical and thoracic spinal cord. He did well, though, for a number of years on rituximab, a B-cell depleting therapy. But then about a year and a half ago, presented to an emergency department with new onset psychosis. He had no history of psychiatric disorders. And this was about a month after returning from a vacation. He did not have a fever, he did not have a stiff neck, didn't have a rash. But because he was immune suppressed, he had a spinal fluid exam, which was basically normal. He had a mildly elevated protein, but that was it. He was tested by a multiplex PCR panel for a number of viral and other types of neurologic infections, but this workup was unrevealing. He also had an MRI, which was stable. And so it was thought that this presentation was not likely due to his underlying neuroinflammatory disease. And then it turned out that he had new polysubstance abuse with alcohol, methylphenidate, and opiates. And so the thought was that separate from his neuroinflammatory disease, polysubstance abuse was at least partly to blame for these new psychiatric symptoms he was displaying. So he followed up with psychiatry, but then it was noted that he had ongoing cognitive decline over the next few weeks, which prompted another MRI and spinal fluid exam. This time he did have frank inflammation in the spinal fluid with an increased number of immune cells, and his MRI showed new diffuse cerebral atrophy, as well as kind of more confluent white matter changes in the insula, in the anterior temporal lobes here. And this was all not consistent with his underlying demyelinating disease, and really increased worry that there was a chronic infectious process that was superimposed on his original condition. So we sent off a number of, again, pathogen-specific tests that were negative. But then deep sequencing detected a Coxsackie virus B5, an enterovirus. And notably on that holiday he'd had about a month before his neurologic symptoms, he and his girlfriend had a diarrheal illness. So it's possible he may have contracted this enteroviral infection on his vacation. Patients have been reported before with chronic enterovirus encephalitis. Again, we typically associate enteroviruses with more acute neurologic infections, but in immune suppressed individuals, they can form very severe chronic infections. And that's seen with a number of other types of neuroinvasive viruses listed here. Fortunately, the patient responded to intravenous immunoglobulins and use of an investigational anti-enteroviral agent, and over a number of months has made quite a remarkable recovery. So these cases highlight that that direct detection of viruses can lead to clinically impactful diagnoses. But we also know that some viruses, especially in immune competent individuals, can linger in the spinal fluid for very short periods of time or at very low titers, making it difficult to detect viruses in the spinal fluid. And so for that reason, many neuroinvasive viruses are diagnosed by serology in the spinal fluid. And in addition to testing for specific viruses with specific antibody tests, there are new technologies to allow for more, again, hypothesis-free antibody testing for viruses in CSF and other bodily fluids. And this work was started largely by Steve Elledge's lab at Harvard and is being continued by Ben Larman, who worked with him on this at Johns Hopkins. And they use a technique called programmable phage display, in which DNA sequences of interest are designed and then synthesized and then cloned into the bacteriophage genome so that peptides encoded by those DNA sequences can be displayed on the surface of the bacteriophage. Then those bacteriophage can be incubated with patient sample, in our case, spinal fluid, and then a number of immunoprecipitations can be done to pull out the bacteriophage displaying cognate antigens for the patient antibodies, and through sequencing those sequences can be identified, and you can assess for log fold change above background. And so as a nice proof of principle of the role of this technique for diagnosing neuroinvasive viral infections, Dr. Nath, who's who's co-chairing this panel with me today, worked up a case with Ben Larman of a patient with a relatively rapidly progressive dementia who's originally from India, and who'd had a massive diagnostic workup without a diagnosis. But VIR-scan detected antibodies to dengue virus at high levels relative to blood, and suggesting that this typical, what we typically associate as a acute infection caused by dengue virus, that dengue virus might also be able to cause a chronic neurologic infection. And they were able to show that after this patient unfortunately died, that the brain was riddled with dengue virus RNA, suggesting that there really was a parenchymal infection with this flavivirus. And then we similarly have adapted and used this technology. We've created our own VIR-scan library with a larger number of viral peptides, but conceptually the same idea. This is a library that has peptides tiling across all vertebrate, tick, and mosquito viruses. And we utilize this tool to investigate another neuroinflammatory syndrome, acute flaccid myelitis, which is a polio-like illness afflicting children. It's been increasingly common in the US in the early to mid 2000 teens. It's become less common now during the pandemic. And this polio-like illness, for epidemiologic reasons, has been thought to most likely be due to two different enteroviruses, D68 and A71, but it's been difficult to identify the virus within the spinal fluid itself, leading to questions about whether there could be some alternate additional infections leading to this syndrome of acute flaccid myelitis. And so we, collaboration with the CDC and a number of hospitals around the country, assess spinal fluid from these children relative to children with other neurologic disease controls. And similar to a study published around the same time for me and Lipkin's group at Columbia, found that these children had elevated levels of enterovirus antibodies and not to any other family of viruses that were represented in the VIR-scan library in their spinal fluid. Again, providing additional circumstantial evidence that the epidemiology is correct, that it is likely enteroviruses that are implicated in these cases of acute flaccid myelitis. Then I want to highlight another way in which we think that viruses can lead to neurologic disease. Of course, they can be, the most obvious way is the virus itself can be neuroinvasive, and we can detect that either through direct detection by culture or nucleic acid detection, indirect detection with serology, or in cases where we think that the virus itself may trigger an immune response and cause a para-infectious neurologic phenomenon, we can look for evidence of autoantibodies. So this is a case of a patient, 30-year-old man who was previously healthy, that we evaluated together with our colleagues at Yale, Drs. Shelley Farhadian and Serena Spudic. And this man, day after his COVID-19 diagnosis, developed a delusion that the rapture was imminent. And this delusion persisted over a number of weeks and was refractory to inpatient psychiatric hospitalization and antipsychotic medications. 5 weeks into this illness, he was admitted to the medical hospital at Yale and had a neurologic evaluation, which was largely unrevealing. But because there was a worry about a para-infectious etiology, he was given immune modulatory treatment, IVIG, and had a very dramatic improvement in his symptoms within a day. Because of that therapeutic response, in collaboration with my close colleague Sam Pleasure at UCSF, and a talented postdoc, Chris Bartley, we looked at the spinal fluid from this patient to see whether there was evidence of anti-neural antibodies, and found that when we incubated the spinal fluid with rodent brain tissue, as you can see here, that there was evidence of immunostaining in a variety of anatomic regions, and specifically to neurons. suggesting that there was an anti-neural antibody present in the CSF at the time the patient was ill. Again, we don't know if these antibodies are at all pathogenic, but it was an interesting observation, especially in light of his apparent response to IVIG. We and others have published other papers in patients, rare patients with neurologic complications of COVID-19 who had evidence of inflammation in the spinal fluid. Notably, it's been very difficult to identify the SARS-CoV-2 viral RNA in spinal fluid and brain tissue. Dr. Nath's group has done extensive work on this. So it opens up the possibility that these in rare patients, that neurologic complications may be instead due to para-infectious causes. So these are some other cases, again, that we evaluated with colleagues at Yale with evidence, again, of anti-neural antibodies, different patterns in different patients with different neurologic phenotypes. And we've done some additional antigen discovery work in these cases, and in some cases identified specific autoantigens. Again, whether these autoantibodies are pathogenic or not is not known, but these have been interesting observations in specific cases. And the last part that we've been working on is generating monoclonals from expanded B cells in the CSF of these patients, and finding that with some of these monoclonals, there's evidence both that they target autoantigens and that they have cross-reactivity to different regions of the SARS coronavirus 2 spike protein. So I'll conclude there and just highlight that viral discovery in neurologic conditions, especially patients presenting with acute or chronic encephalitis, is a really important area of research, given the number of emerging and re-emerging viruses that have neuroinvasive potential. Deep sequencing is a very useful tool for this type of work. In addition to identifying a virus, it also can allow for more detailed phylogenetic work to determine the exact source of the infection. But it's also important to recognize that metagenomic sequencing has real limitations. If the infection is compartmentalized, if it's a virus that is present only for short periods of time, if it's very low abundance in the CSF, or if there's high human background because of a very high cell count in the CSF, these are all reasons why metagenomic sequencing may be insensitive. I didn't talk about this today, but there's work done by us and others suggesting that the host transcriptomic data coming out of the metagenomic assay can be useful for classifying patients with viral versus autoimmune causes of encephalitis, for instance, and that work is ongoing. And then lastly, we discussed how broad-based viral antibody profiling can shed additional light on potential neuroviral infections, although techniques like phage display do have some limitations, including being insensitive for detecting conformational epitopes or antibodies targeting post-translationally modified regions of proteins. This cited a number of studies along the way, which have been highly collaborative, and I want to thank our collaborators here as well as our funders. Thank you very much.
Thank you, Michael, for that terrific presentation. So, so you mind taking a few questions now? So as questions come in, so one thing I was wondering is, what about false positives with next generation sequencing? Is that a concern ever?
Absolutely. So I think it's less of a concern when it comes to virus detection, but it's not, the many environmental contaminants that we see in sequencing data are from bacteria and fungal flora on the skin or in laboratory reagents, but there are some well-known cases of viruses that have been detected and initially suspected to be, disease related that turned out to be contaminants from the lab. There's a case of XMRV that was initially thought to be associated with a prostate cancer that turned out to be a lab contaminant. There's I believe a PEGI virus infection that was thought to be associated with a viral hepatitis in patients who were seronegative for kind of known viral hepatitides. That similarly turned out to be a reagent contaminant. So absolutely, it can happen, and we've had that. We've gotten excited before about unusual viruses that turned out to be, contaminants in QIAGEN kits, et cetera. So I think, this technology doesn't replace kind of needing to clinically correlate, what's going on with the patient. And then also, the need for really good negative controls, on all these experiments, both to know kind of what common contaminants are, but also kind of time updated controls, because even though many contaminants are usual suspects that you see over months and years, with a new lot of tubes from not to pick on QIAGEN, but from QIAGEN or another vendor, there may be a contaminant that just hasn't been seen before. So yeah, it is a real concern.
Okay, we have a few questions here in the chat. So first is from Jamie, and says that you mentioned that in most cases of SARS-CoV-2 has not been found in CSF or brain biopsy. Do you think this is due to difficulty identifying it, or the fact that neuroinvasion is less important in manifestations of neurological symptoms in COVID-19?
So I'm sure, Avi, you have very detailed thoughts on this. I can give my 2 cents, but you're the real expert. My sense is that like you've looked probably the most deeply at this, is that it may just be that viral neuroinvasion is less of a factor to account for the variety of neurologic complications that we see in COVID-19. Because I think that your work and others, you've looked very deeply in many postmortem brain tissues, and at this point, I think if it was there, that it would have been found more consistently. So to me, it really suggests that a lot of the neurologic complications are due to kind of systemic disease from COVID-19, and in some cases potentially kind of peer-infectious immune mechanisms. But I'm curious to hear what you think too.
I agree that, for example, if you look at various types of viral encephalitis, let's say rabies or herpes or something, they're just loaded with virus. You'll find it all over the brain. So here, even those that say that they found some virus, they find very, very small amounts of it in the brain. So, but if you look at the pathology, it's pretty widespread. So that small amount cannot really explain the pathology. So there has to be other immune-mediated mechanisms, I think, that are important. Okay, we have another question, last one, and then we'll. So this says that VirScan seems like a sensitive method to scan known viruses present in the environment. Could it be designed so that you can detect novel viruses?
So I think that it. Well, for instance, we kind of did that experiment. We when SARS-CoV-2 first came on the scene, we had our existing VirScan library, which of course did not include SARS-CoV-2, but included other known human coronaviruses, and although it and we were certainly able to detect antibodies targeting SARS-CoV-2 with that library. So I think absolutely there's a lot of many parts of viral genomes are relatively conserved across that family of viruses, and so certainly for detecting antibodies that target conserved regions of the genome, a large display library like this with tens or hundreds of thousands of peptides should be able to pick up a signal. It may not be as good as a dedicated library for that specific virus, but certainly should be able to detect an previously unknown relative.
Thanks. Michael is also the co-chair of this session, and Michael, do you want to introduce Dr. Klein?
Absolutely. So So it's my pleasure to introduce Dr. Robyn Klein, who we're very lucky to have with us here today. She's the Robert E. and Louise F. Dunn Distinguished Professor of Medical Sciences at Washington University in St. Louis. There, she's the director of the Center for Neuroimmunology and Neuroinfectious Diseases, and a professor of medicine and pathology and immunology and neurosciences. She's also the vice provost and associate dean for graduate education in the Division of Biology and Biomedical Sciences. In her laboratory has really been a leader in defining novel roles for cytokines and chemokines in the regulation of blood-brain barrier permeability, to specifically working extensively on arboviruses and defining the mechanisms of neurologic sequelae of these neurotropic neurovirulent viral infections. So it's a real pleasure to have you here today. Thank you.
Hi, everyone. I'm Robyn Klein. I'm coming to you from Washington University School of Medicine in St. Louis, Missouri. And I want to thank the organizers for inviting me to present my work. In these 15 minutes, I'm going to talk to you about some work we've been doing to look at virus-mediated cognitive impairment. And so in my lab, we study emerging and re-emerging RNA viruses that can induce acute and chronic neurologic diseases. Here I'm just showing you West Nile and Zika virus, which are both members of the Flaviviridae family, and they are transmitted to humans by mosquitoes. More recently, we've also begun looking at the effects of neurovirulent viruses in the brain, including SARS-CoV-2, which, as you know, is a coronavirus that is transmitted through the respiratory route. So to begin with, flaviviruses are enveloped positive single-strand RNA genome viruses, and they're expressed as a single open reading frame. Some of their non-structural proteins have various effects on the blood-brain barrier and also on innate immune binding. West Nile virus is the leading cause of encephalitis in the United States, and it can cause not only encephalitis, but flaccid paralysis, meningitis, and can lead to long-term to cognitive impairment. West Nile is deposited in the skin by a mosquito, where it may be taken up by Langerhans dendritic cells and where it spreads to local nodes. There's a short-lived and low-level viremia, and then the virus in some patients can spread to the central nervous system. It can spread via retrograde axonal transport or cross the blood-brain barrier. Once it gets into the central nervous system, it targets neurons and only neurons. In terms of the initial innate immune response to flaviviruses, these viruses are detected initially by three classes of pattern recognition receptors. There is the toll-like receptors shown on the left that can sense viral double-stranded or single-stranded RNA. There are the retinoic acid inducible gene-like receptors, RIG-I and MDA5, and some of the flaviviruses have evolved mechanisms to avoid recognition by these sensors. There's also the NOD-like receptor family Pirin domain containing protein 3, or the NLRP inflammasome, which can also sense virus, and virus will bind these receptors and induce inflammasome activation. So in terms of the pattern recognition receptors, these will lead to the expression of lots of inflammatory genes, including type 1 interferons shown here, which will bind to its own receptor in a paracrine or autocrine fashion and lead to the upregulation of pro-inflammatory molecules, including chemokines that will recruit T cells that enter the CNS to clear virus. And this can be done non-cytolytically. Since West Nile emerged in 1999, there has been multiple studies demonstrating that more than half of patients that survive either neuroinvasive disease or even West Nile fever can represent with memory impairments. And this is also true of Zika virus. I'm showing you some data here demonstrating a study where there were neurocognitive findings in patients with a history of West Nile virus neuroinvasive disease. This was published by a colleague, Christy Murray. And what you can see is that visuospatial learning especially is impaired in these patients. So spatial learning is very well established in mice. And so we developed mouse models to look at post-infectious cognitive recovery from West Nile or Zika virus, where we inoculate the viruses intracranially. There's peak infection is cleared. And I'm showing you on the bottom that we've been using an attenuated strain of the virus, which is called West Nile virus NS5E218A. It has a single amino acid substitution in the non-structural protein 5. And that it can lead to about 90% survival in mice. It can lead to very similar viral loads shown by these plaque-forming units per gram of tissue, as opposed to the New York strain, which we've always used in the past, which gets inoculated via the foot pad and leads to high variability in whether the CNS becomes infected. So we've been using behavioral tests to look at post-infectious cognitive recovery. In a paper we published a few years ago, we used the Barnes maze. We also used open field testing. I don't really have time to talk about how these work, but suffice it to say that we found that West Nile virus recovered animals displayed significant numbers of errors and difficulty locating a target hole on the Barnes maze. However, didn't display any difference for mock infected mice in terms of crossing lines in a center box, suggesting that this was not an alteration in anxiety, but actually a spatial learning defect. So over several papers, we demonstrated that there were mononuclear cell triggers that impacted learning and memory with effects on integrated systems. So while West Nile virus entered the central nervous system and Zika, CD8 positive T cells that are directed and antigen specific at virus enter the CNS, and through type 2 interferon or interferon gamma can clear virus, but they can also lead and promote activated microglia, which through expression of complement can tag synapses for elimination and lead to spatial learning. We also found that macrophages would deliver IL-1 beta to the brain. And although they retreated, IL-1 beta levels stayed high. And what we observed was that there were loss of neurogenesis in the hippocampal formation under the dentate gyrus, which is an important area for neurogenesis that's related to spatial learning. And the people that discovered all of these things are shown here: Mike Vasek, Cherise Garber, and Allison Sung, who all have gone on from my lab to do other things. And so one of the questions that remained that has been recently published in a paper in Brain Behavior and Immunity, so although it says in revision, it's actually in press, is that astrocytes can have inflammasome activation. One of the questions I repeatedly got was, how was IL-1 beta being expressed by astrocytes, even though they're not targeted by virus? So in the top, I'm showing caspase-1 activity in astrocytes and myeloid cells at 7 and 25 days post-infection. You can see that the myeloid cells are in fact delivering inflammasome cytokines such as IL-1 beta through caspase-1 activity early on, and then by 25 days post-infection, you can see that astrocytes now take over this process. And in B, I'm showing that many of the inflammasome complex components, such as picard and Cas1 and LRP, are all expressed by astrocytes only in the Western Nile and virus recovered state. And you can see caspase-1 co-localization with GFAP expressing astrocytes, and that this increases over time, and that NLRP3 is also co-localized with astrocytes over time after recovery from infection. So in trying to discover which cellular target of IL-1 can limit synapse repair, we had to deal with the fact that all of these cell types shown here all express the IL-1 receptor. And so we attempted to use Cre-Lox technology to identify which cell type was the most important and the most, the target for IL-1 beta in our system. And so we bred nestin Cre, ALDH1L1 Cre, CX3CR1 Cre, and CAM kinase 2 Cre lines to our IL-1 receptor flox mice. And I will cut to the chase and let you know that the only line that actually demonstrated any effect was the nestin Cre line. And so loss of IL-1 receptor 1 in neural stem cells could restore neurogenesis, shown in the top. At 52 days post-infection, there's still loss of neurogenesis in animals that express the IL-1 receptor on the nestin positive cells, whereas in those where it's been deleted, have restored neurogenesis. We also saw that synapses that were eliminated in the brain of animals that recovered from West Nile could be restored and also prevented from being removed when we deleted the IL-1 receptor 1 from their neural stem cells. So many of you are aware that astrogenesis is regulated by STAT3. So neural stem cells can go two pathways. They can go down a Wnt signaling pathway to make neurons, or a JAK-STAT signaling pathway to make astrocytes. And so we performed bulk RNA sequencing of adult neural stem cells that we harvested from normal hippocampal dentate gyrus that were treated with vehicle versus IL-1 beta. And what we found was that there were a number of upregulation in the IL-6 JAK-STAT3 signaling pathway, suggesting that IL-1 beta really causes a gliogenic switch in these adult neural stem cells, so that they derail neurogenesis in favor of astrogenesis. And on the bottom, I'm just showing you that the Cre-positive mice, where the IL-1 receptor 1 is deleted from neural stem cells, does not express any phospho-STAT3, which is clearly found in the subgranular zone in the Cre-negative animals. We also found that our Cre-positive animals had a restoration of loss of astrogenesis, and also we found that the GFAP expressing IL-1 beta astrocytes were no longer being generated when we deleted this from the neural stem cells. And then finally, we were able to rescue spatial learning defects. So shown here, the Cre-positive animals in the blue squares, their learning curves were very similar to the mock infected animals of both Cre-negative and Cre-positive mice. And we had also shown previously that if you administer an IL-1 receptor antagonist during the acute phase of infection, and then look 52 days later at spatial learning, you could prevent the development of spatial learning defects. And so adult hippocampal neurostem cells respond to inflammation and maintain inflammation through the effects of IL-1 beta on neural stem cells. So what about neurovirulent viruses? Can they trigger this process? And so I'm just going to show you two slides, some work that is being done in my lab looking at SARS-CoV-2 and effects on the brain, particularly in the hippocampus. So this is just some immunohistochemical data showing in the top that hamster hippocampus over various time points post-infection with SARS-CoV-2 increases its level of IBA1 positive cells, and that these cells are actually making IL-1 beta, and that in human COVID cases, we also found that IL-6 was expressed by neurons within the hippocampus. So two anti-neurogenic cytokines are being made in the SARS-CoV-2 infected brain in animals and humans. And then just to show you effects on neurogenesis, we found in both hamsters and in humans that acutely infected animals and patients led to loss of proliferating neuroblasts in the case of the hamsters, which you can see down here on the bottom graph, where there's double Cortin labeled cells with Ki67 labeling, and that there were decreased numbers of these neuroblasts in humans with COVID-19, and also decreased numbers of immature neurons within the granule cell layer, suggesting that neurons are not integrating into the circuit in patients that are acutely affected with SARS-CoV-2. So as far as conclusions, I think for this part of the talk, I've shown you that we have a hamster model. I didn't have time to show you blood-brain barrier disruption, which was also found in human cases, and that this model can model human COVID-19 in terms of brain cytokine expression and loss of neurogenesis. And so it would appear that anti-neurogenic cytokines that are expressed by microglia, maybe neurons, and astrocytes in the case of flaviviruses can reduce the numbers of neuroprogenitor cells and neurons. And so there's always the question of whether we can repurpose anti-IL-1 or other biologic drugs to prevent post-infectious cognitive dysfunction due to viral infections, and whether this would work for neurodegenerative diseases caused by protein aggregates, which are also detected by PRRs and activate the inflammasome. I just want to thank everybody in my lab. I'm grateful for my funding and collaborators. I'll stop now.
Great. Thank you so much, Robyn, for a wonderful talk. And we have one question already. Is there a relative preference for cognitive deficits versus other neurologic or psychiatric sequelae in the viral infections you discussed? If so, does this imply relative vulnerability of specific brain regions, especially hippocampus, to neurotropism? And if so, why are these regions more vulnerable than others?
So that is a fantastic question. And certainly with the flaviviruses and even with COVID-19 cases, there are other psychiatric sequelae that are well known. But I'd like to sort of mention that there's been a longstanding history of folks determining that the hippocampus has actually a unique susceptibility to exposures. And this not only includes viruses, but includes toxic agents. And so it's really unclear why that's true. We do find that the hippocampus tends to have a higher permeability at the blood-brain barrier. It may be because of its proximal location to the ventricles. And so there may be more cytokine expression that's found as cells can actually exit into that compartment, and it's possible that that's part of it. But I don't think we really know completely why there is more sensitivity, but that's a really good question.
I had a question, just a lot, as you've published extensely on viruses that have a clear neurotropism, like West Nile and Zika. And have you, with SARS-CoV-2, where we think of it primarily as a respiratory virus, have you looked at other respiratory viruses, primarily respiratory viruses in the past, and do you have a sense that that they similarly can have effects in hippocampal function, or is this something you think that is really unique to SARS-CoV-2?
So I haven't looked at other respiratory viruses in animal models, but I'm been starting to collaborate with somebody who works on high pathogenic flu viruses. But H1N1 is certainly a virus that has been associated with neurologic effects that are similar to what you see in SARS-CoV-2. So it doesn't appear that there's a specificity, but it may have something to do with a level of inflammation systemically or other effects that respiratory viruses can have on trafficking of cells and barrier effects. So I think we just know this much at this point.
Okay. And then we have another question here. Thank you very much for the excellent presentation. During the chronic inflammation after the virus has been eliminated, if you were to treat animals with drugs such as dexamethasone to dampen the immune response, could that improve spatial learning and regain adult neurogenesis?
So we haven't tried that specifically. I think that what we have found is that certainly in the flavivirus neurologic sequela, that resident memory T cells appear to be the most proximal trigger of microglia. And so if you were able to somehow use lympholytic agents, steroids can eliminate cells in the periphery. It's unclear how well they can get into a normal CNS and do this once folks have recovered. But it's certainly something that we're looking at in the laboratory.
Great. Thanks so much.
Okay.
Okay, so I'm going to introduce the next speaker here. So this is Dr. James Badenoch, who's an academic foundation doctor at Barts Health NHS Trust and Queen Mary University of London. He's a member of the SARS-CoV-2 Neuro Collaborative Network. He collaborates on a weekly blog of research on the neurology and neuropsychiatry of COVID-19, And the title of his talk is Persistent Neuropsychiatric Symptoms After COVID-19, a Systematic Review and Meta-Analysis.
Hello, my name's Jamie. I'm a junior doctor working in East London, and today I'll be presenting some research exploring the neuropsychiatry of long COVID. So this research started with a blog which evaluated nervous system complications in COVID. And quite early on through doing this blog, we found evidence of persistent neuropsychiatric symptoms in people that had recovered from the acute phase of the illness. So we wanted to assess the prevalence and potential driving factors of these symptoms through a systematic review and meta-analysis. This is important both on a theoretical level to improve our understanding of what long COVID actually is and also what drives it, and also to get an idea of how many people are affected so that we can adequately plan for services that will be required. I'll now talk you through a graphical summary of our project. So we included 51 studies, nearly 20,000 participants, and on average, they're about 3 months after initial COVID infection or after hospital discharge. Most studies only looked at individuals who were hospitalized for COVID, and the majority were done in China, Europe, and North America. In terms of quality, the majority of evidence was drawn from cohort studies of medium quality without controlled populations. Now let's get to the actual symptoms. The most common in descending order was sleep disturbance, fatigue, cognitive dysfunction, anxiety, and PTSD. In terms of potential driving factors, we wanted to know how these symptoms changed over time. Concerningly, we found no significant difference between symptom prevalence before and after 3 months following hospital discharge. This could indicate that these symptoms do not wane during this initial period following hospital discharge. And furthermore, no significant difference in symptom persistence was found according to COVID disease severity, and proxies for disease severity were being hospitalized or requiring critical care admission. So the key points I'd like to draw out are that persistent neuropsychiatric symptoms after COVID are common, they're not limited to that initial post-acute phase, and they're also seen in those with more mild or moderate disease. So we were thrilled to have the paper accepted by Bain Comms, and it's currently on their website. And I'd just like to end by saying this project was very much a group effort by a research group set up during the pandemic. We've now got a dedicated website and a Twitter page where we host our blog, our database, and ongoing projects that I encourage you to take a look at. Thank you very much for listening.
Thank you, Jamie. So we'll take a second here while questions are coming in. But I'm just curious about where we go, where you're going next with this work, what the kind of future directions are for delving deeper into these issues.
Yeah, I think that's a really interesting point. So our paper, the literature search was actually done about this time last year. So it was quite early on in the long COVID literature base. And it was mainly just to get a scope for what kind of evidence is out there. There hadn't really been any systematic evaluation of the literature at that time. And since then, a few different things have come out. So at that point, there was a real dearth of kind of prospective and longitudinal studies. And since then, there's been a bit more evidence to kind of suggest to explain the scope of neuropsychiatric symptoms, but also to show, for instance, the TACE study and the FOSP study, which have looked at large numbers of people from TriNext database and also from people that were hospitalized in hospitals across the UK, to show that some of the driving factors that we didn't find any evidence for, such as disease severity and hospitalization, were actually significant driving factors for persistent neuropsychiatric symptoms and for a diagnosis of a psychiatric disorder 6 months after initial COVID infection. So it likely shows that probably at the time when we did the meta-analysis, because of the relative poor quality of the literature, that a lot of our analyses were underpowered. But it's been interesting to see, potential driving factors for these symptoms and overall to demonstrate on population level how significant the psychiatric sequel of COVID can be for many.
Thank you. And then Avinath was wondering, do you see any patients with functional neurological manifestations in your series?
Yeah, I think it's a really interesting question. Within our scope, we didn't include FND as one of our outcomes because we based our neuropsychiatric definitions on patient-led terms, because it was kind of patient-led research initiatives that started a lot of the long COVID literature. So we thought it was only fair to kind of put it within patient terms. And FND didn't fall in with it. There has been definitely an increased rate of FND, I think, after vaccinations, and there's been quite a lot of literature demonstrating that, but I'm not actually sure whether there's been evidence looking at things like on a populational level, whether they've increased during the pandemic, to be honest.
Great. Well, thank you very much.
Thank you.
Okay, should we move on to Tom Solomon's talk? Thanks, Michael. So I'll take.
Thank you very much for inviting me to talk at this brain conference. My name's Tom Solomon. I've been asked to talk about emerging challenges in brain infections. I'm based at the University of Liverpool. In terms of disclosures, I sit on several government advisory bodies around COVID-19. I'm the president of the Encephalitis Society, and I'm the vice president of the UK Academy of Medical Sciences. So I've been asked to talk about emerging challenges. Last year in this same conference, I was asked to talk about emerging causes in brain infections. So this time I've broadened it out to some of the wider challenges, and it's given me an opportunity to scratch my chin and think a bit about the future. And interestingly, I gave a talk a couple of years ago on encephalitis, the next 25 years. And it's quite interesting to see which of those predictions have come true just in the past couple of years. And I'm basically going to be talking about new data on emerging causes, how we need to improve diagnosis, and then a little bit about better treatments. And if I might quote from Steve Jobs, you can't understand what's happening today without understanding what came before. So I'm going to look a little bit at what we've done in the past to think about how that might inform the future. And my past working on brain infections begins in 1994, which is nearly 30 years ago, in Vietnam, southern Vietnam. And I've continued to work in Southeast Asia, and then in Africa, and then also in Latin America and Brazil since then. And I'm the director of the UK's Emerging Infections Research Unit, which has given us the opportunity to work on some of these emerging infection problems. And this is quite a nice map the UK government produced showing the emerging infection events between 1998 and 2018 when it was produced. And you can see most emerging infections are zoonotic, so they've spread from animals to humans, and many are transmitted by arthropods, mosquitoes or ticks. And in green, they've covered newly identified infections. In brown, those that were known about already but have spread to new areas. And then in blue, those that have emerged and spread. And of course, this was 2018. In 2019, at the end of 2019, we might have put this little blue spot in Wuhan. And by now, the map should really look like this if we wanted to cover COVID-19. So what are the factors that are contributing to emergence and spread of infectious diseases? I've listed some of them on the left there. And you can see in the figure on the right that a couple of issues really. One is the time it takes to travel around the world. In 2000, and since 2000, you can do it in about 24 hours. Back in the 1850s, it took about a year on a ship. And then the world's population has gone up. It's now about, well, it's more than 7 billion now. Back in 1850, there was hardly anyone on the planet. And so these things have undoubtedly contributed. But also there are other things listed down the left there, changing agricultural and farming practices. We move crops and livestock around the world. Humans and animals quite often live in close proximity. Climate change has been important, of course, and then pathogens evolve. But if we think about which of these can we do something about, well, we have shown that we can reduce human travel over the past couple of years, although like me, you're probably looking forward to traveling a little bit and doing meetings like this, where we can actually meet and talk to people rather than talking to a computer. Love it, though I do. But so many of these on the left are things that can change and can be changed, particularly if we adopt this One Health concept, which is the idea that the health of humans and animals and the environment are linked. And we're going to see quite a lot of activity in this area in the next few years. Pathogens, we can't really stop pathogens evolving, unfortunately. It's called Darwinian evolution. Now, some of the important pathogens are arthropod-borne, as I mentioned, and this map just shows you some of the important arboviruses, which wherever you are in the world, there are important arboviruses, even here in the UK. But we, some years ago, thought about which arboviruses might grow in importance, and we published this in Lancet in 2008. And you can see if I make this bit a little bit brighter here, that we talked about flaviviruses, which seem to have a promising future and in many cases aided and abetted by mankind, like I said, the kind of things we do. And we did highlight Zika as a potentially emerging virus back then. And this proved to be one of our predictions, which was correct. Many of them, of course, are not at all. One of my colleagues, Matthew Bayliss, has been leading work seeing what can you predict based on things like climate change, weather patterns, etc. And he's showed how the Zika outbreak, large outbreaks, of course, in Latin America, were probably partly consequent on the El Nino effect, the climate effect, where you get a warm, moist climate. And this happened in 2015 and supported the explosive growth of mosquitoes and then the mosquito-borne virus, Zika. We've also been looking at the impacts of climate change in Europe. And basically, we think that by within the next 30 or 40 years, the southern UK will be warm enough to support vectors like the Aedes albopictus mosquito, which transmits dengue. So you can think a bit about what are we going to see in the future. And in the UK, I think we will see dengue, West Nile virus and Usutu probably. We already have the Culex mosquitoes that transmit these viruses, though not in great numbers. We predicted, so this slide is an old slide from this talk, the 25 years talk I had to give. And we predicted tick-borne encephalitis virus coming to the UK then, and indeed it has come. Oropouche, not particularly a UK problem, but I know that this meeting has a wide audience, and this may well prove to be an important problem in Latin America and then grow from there. So respiratory viruses, we made the comment back then that we were waiting for the next flu pandemic. And then we always include Agent X in these lists, which means the pathogen that you know nothing about yet, but that may just emerge and spread rather rapidly. And of course, that's proved to be the case. Boris Johnson in November 2020 declared that we were going to have some zoonotic hubs to identify dangerous pathogens and predict which might emerge. Unfortunately, he's got rather distracted since then, but we have in Liverpool launched the Pandemic Institute at the end of last year with £10 million of funding. None of this government funding, although the UK government has promised to put funding into this kind of thing. I better pause. Hang on. How do we pause? Right. Apologies. I think we got as far as here before I had that interruption. For some reason, my 18-year-old daughter is pathologically unable to take a key with her when she goes out of the house. So Boris is not the only one distracted. Anyway, we have launched our Pandemic Institute with funding, not from the government, but from philanthropists. And in terms of predicting the future, so again, my colleague, Matt Bayliss, has been doing machine learning work, which integrates mammalian viral and network traits to predict unknown virus-mammal associations. And with these tools, he thinks he can predict what will be our next emerging virus event. But as somebody once said, shit will happen, but remember it will pass. And indeed, I think we are passing at the moment. Let's move on to talk about diagnostics. So this is an old slide, not that old, but it showed how proud we were that we were moving virology diagnostics from the International Reference Lab down to the simple viral kits, such as, for example, this one that we've been using in Southeast Asia, which you can use in hospitals rather than in big labs. But of course, this has all advanced enormously just in the last couple of years, so that now people are using these simple kits at home, which is a great advance. And I think this approach will be used widely now across many other infections. Now, I know Michael Wilson, by the time you hear this, he will have talked all about the metagenomics for detecting pathogens in terms of new approaches to diagnosis. So I wanted to just talk a bit about new approaches examining the host response. So rather than looking at pathogens, we look at the host response. Obviously, antibody detection is a very specific way of looking at the host response. But the other way is a non-targeted way of looking at the host response by looking at the patterns of cytokine genes, or indeed microarray to look at host genetic mRNA production. And here's an example of this from Mike Griffiths in Liverpool, showing that you can use host gene expression to distinguish, in this case, Japanese encephalitis patients from patients with JE mimics. And we've also used this approach actually to distinguish patients with bacterial from viral CNS infections. And this has gone to be patented. And this approach, the same approach or very similar approach used by Mike Levine, has proved useful in distinguishing, this is just any febrile illness, viral from non-viral. So in the next 25 years, diagnostics will be quicker, non-targeted, automated, and very much closer to the patient. Will this spell the end of microbiology? I don't think so, the end of traditional microbiology, but it's just moving into new areas. But what I do think is worth looking at is this. So when I started my career in '94, when we looked at cohorts of patients with suspected encephalitis, we could diagnose about 50% of them. This number has gone down over the years. We're now at about 20%, but we're still very much focused on either detecting the pathogen in the nervous system or looking at the immune response through things like antibody assays. But we've always thought that, you know, a large proportion of patients with encephalitis, the unknowns, we don't think it's because we're not able to find the pathogen. We just basically think the pathogen is not there in the CNS. And it's been our idea for some time now that actually this encephalitis in the brain may be the body's response to a systemic infection rather than a CNS infection. And I think during the pandemic, we've actually seen this to be the case. So you'll be familiar, I hope, with the ideas of how SARS-CoV-2 causes neurological disease. A small amount is from direct viral invasion. Some of it's from prothrombotic events and blocking vessels. But it would also seem that the systemic infection is causing an amount of inflammation in the brain, even though the pathogen isn't getting there. And that is then causing problems like the psychiatric events we're seeing, the encephalopathy we're seeing, et cetera, et cetera. What about machine learning? Well, there's certainly going to be a big push on this. It probably is going to have impacts, but I still think that clinical medicine learned at the bedside, listen to your patient, he's telling you the diagnosis, said the great Sir William Osler. I still think this is going to have a role. And just a couple of examples of this. The Guillain-Barré syndrome associated with Zika virus infection. This was picked up on really because of clinical skills in looking at what's going on with the patient and thinking about that. Similarly, the work in COVID, determining that there are neurological and neuropsychiatric effects. Again, that's the DHL man. You see, this is the problem with these online conferences. If we were all together, and of course we could be now, there are no restrictions on travel now in the UK, we could all be together in London having a proper meeting. Anyway, the work on determining neurological and neuropsychiatric complications of COVID-19 has come again from basic clinical skills, looking at and thinking about patients. So I think artificial intelligence and machine learning is going to, clearly it's important with imaging, it's important with histopath. It probably will contribute to what we understand clinically with big data sets, but I think there will always be a role for clinicians who can look at patients and have a think about what's going on. So let's think now a bit about treatment of encephalitis in the future. And we can divide this into thinking about antiviral drugs, ancillary treatments, and then also thinking a bit about clinical trials, etc. So first of all, the antiviral drugs, there is scope clearly for discovery of new drugs, rational drug design based on what we know about the viral properties. So here's an example of antiviral inhibitors targeting the NS3 helicase protein of Japanese encephalitis virus, built on similar work for hepatitis C. I wanted to say a bit about treatment trials. Moving on now to think about herpes simplex virus. So many of you will know about the DEX-ENCEPH study, which is a trial of dexamethasone in herpes simplex virus encephalitis. The target was 90. As you can see from the red line there, we've actually gone above the target now. We're at about 93 patients, and the study ends at the end of February. And we will be looking at the final outcomes in 6 months' time. So we should be reporting, we hope to report the outcome of the study in the December meeting of the Encephalitis Conference in London. And then this is our new study, which started a couple of months ago, ENCEPH-IG. It's a trial of intravenous immunoglobulin in clinically diagnosed patients with autoimmune encephalitis. We're asking the question whether starting IVIG at the same time as you start steroids in these patients will improve the time to recovery. And we are open to new centres joining us. We have 5 centres so far across the UK. We're recruiting patients. And if your centre is interested in joining, or you'd like to know more about the study, please get in touch. And then when we're talking about clinical trials, there is clearly much more that can be done. This is Japanese encephalitis. The total number of patients that have ever been entered into trials of JE are 677, only 381 of whom had confirmed JE. So there clearly is scope for more to be done. And I think we've seen with things like the ability to rapidly recruit patients into COVID studies, not neurological COVID, but broader COVID disease, the recovery trial, then we really, I think, do have to put more effort into these types of treatment trials to answer critical questions. And then the other area where I think there's massive scope for improvement globally is just improving the basic management. We have a program ongoing at the moment called Brain Infections Global, funded by the NIHR, to just see, can we get the basics right? And as anyone working in any setting knows, even in high income countries, this often is suboptimal. So we're looking at really, can we improve things like disease recognition, better early investigations, improve the diagnostics, and then get people on the right treatment. Many of you will know that the World Health Assembly announced in November 2020 the Global Action Plan on Epilepsy and Other Neurological Disorders. And I mentioned at the start that I'm the president of the Encephalitis Society. This has now been going for over 25 years. And just before the pandemic started, I should have mentioned World Encephalitis Day is 22nd of February every year. We'll have had this year's by the time we have this conference. But I did want to mention, just before the pandemic started, myself and the chief executive of the society, Ava Easton, were in Geneva talking to the World Health Organization about our desire for a much more concerted effort globally on tackling encephalitis. And this is now going to result in encephalitis roadmap. There will be meetings on getting this started over the summer to see if we can significantly reduce the global burden of encephalitis through better prevention, diagnosis and treatment, and to a large extent using what we already know. I'm going to stop there. I'll just mention many of you will have been at our COVID neuro webinars over the last 2 years nearly. We're now morphing these into the clinical exchange program, which is about a whole manner of brain problems of importance globally. And then also the COVID neuro webinars are becoming the neurological infectious diseases webinars, where we'll be covering a range of different neuro ID topics once a quarter. And finally, if you like something, if you like your science a little bit more lighthearted, this is the Scouse Science podcast, which we do once a month. You can find it in all the usual podcast places. I'll stop there. Thanks very much for your attention.
Wow, that was quite a tour de force, and we have Tom Salmon joining us, who's going to participate in the Q&A after our final talk starting now. This last talk will be given by Dulcey Sarmiento, who's a specialist in internal medicine with a master's degree in neurosciences at University of the Andes, Merida in Venezuela. Title of the talk is Neurological Manifestations of Patients Diagnosed with COVID-19 in a Hospital in Latin America. So thank you.
Good morning to everyone. My name is Dulcey. I am in charge of Los Andes University. We are going today to review this study. So let's begin. Neurological manifestations of patients diagnosed with COVID-19 in a hospital of Latin America. Background: the COVID-19 pandemic has generated a great burden on society and health systems within its conglomerate of clinical manifestations. Neurological manifestations are one of the least mentioned in this regard, but these represent a large component of associated disability. Methods: descriptive observational study of a cohort of 658 patients diagnosed with COVID-19 admitted to hospital areas during the period from January to June 2021, where the neurological manifestation described. Results: neurological manifestations were classified into three categories: central nervous system manifestations, like dizziness, headache, altered consciousness, acute cerebrovascular disease, ataxia, and seizures; peripheral nervous system manifestation; and muscular manifestation. They were divided into three age groups: under 40 years, between 40 and 60 years, and those over 60 years. The largest group of patients was made up of the male gender, and the group older than 60 years was the more affected by these clinical manifestations. Neurological manifestations were present in 84% of the patients. Discussion and conclusions. Neurological manifestation had been rejected in COVID-19 patients, being these of the most disabling and limiting in terms of functional indexes. The long-term consequences of these affectations are unknown for the moment. We recommend that the group carry out a follow-up long-term and thus assess the consequence of these alterations. Thank you very much.
Well, thanks very much. They were both terrific talks. I see that Tom is trying to connect, right? So as he is, I guess we can, and is Lewis available? None of them are? Well, if they're not available, then maybe Michael, you and me now, we can discuss things. I did have a question for you, and that is, you know, and maybe this is in part related to Tom's talk as well. That's what stimulated interest, that there are a lot of other potential pathogens on the horizon. And so what is your feeling for there now? All right, perfect. You joined at just the right moment. Enjoyed your presentation. It was superb.
Thank you, Tom.
Thanks.
A lot of ground. You seem to be in a really good place, though.
I'm not in a good place at the moment. I'm in a car park.
Oh, good. Well, this must be a bit challenging, but it's good. Thanks very much for doing it. So I was just discussing with Michael, which is probably quite relevant to you, is that, you know, you've had a whole slew of pandemics, and what do you think is on the horizon next? Do you think these SARS-CoV-2 related coronaviruses are just going to keep re-emerging? Are there other pathogens you think we should be worried about that are next on the list? Where are we headed?
Well, I mean, the first thing to say is it's impossible to know. And I think it was Metro-Goldwyn-Mayer.
You knew everything.
Said, no. Metro-Goldwyn-Mayer said only a fool would make predictions, but and almost everything I predict turns out to be wrong. So having said that, if you still want me to give an answer, then I think we're still due a flu pandemic. Just because we've had SARS, that doesn't mean we won't have a nasty flu pandemic. You know, we had 2009, which turned out to be relatively mild, but there's no reason why we shouldn't have a reassortment of flu viruses, which produces a very nasty, severe flu pandemic. In terms of other SARS coronaviruses, I mean, I suppose one of the things that occurred to me and others when this one happened was, you know, why has it taken so long for a pandemic like this to happen? possibly, as you may know, there's some data that suggests the last SARS pandemic that really did spread around the world was the Russian flu, so-called Russian flu of 1890s. So I doubt it's very hard to predict, but and also, of course, we're likely to get new variants of the current SARS coronavirus too, which, unless we vaccinate the rest of the world properly, will continue to trouble us.
Just piggybacking on that, I'm just curious how, for Nipah virus in particular, how high up on that is on your radar in terms of potential for spread. You know, given that it keeps, you know, these local outbreaks keep occurring.
Yeah, thanks, Michael, and good to see you. Good to see you, and
Good to see
Abby.
you.
Yeah, so Nipah, you know, as you know, and many colleagues will know, first caused problems in Malaysia, and now it seems to be more prevalent in places like Bangladesh. I think it will cause outbreaks. I don't think it will cause major, and of course, you know, touch wood, and as I've said, it was very hard to predict. But the difference is that Nipah has not really adapted itself so that it spreads from human to human through the respiratory route. So what we tend to see is that most of the people have been infected directly from the zoonotic reservoir. Unless it changes a lot when it enters humans and suddenly becomes much more able to transmit between humans, and then also much more able to, much less likely to cause severe disease. I think one final important message for anyone thinking about these viruses is that what viruses which are very severe don't tend to cause pandemics because you can spot them easily. That's the difference between SARS-1 and SARS-2. SARS-1, severe disease, people get very sick, they don't get on airplanes and travel around the world. And SARS-2 was the perfect combination of not causing such severe disease in most people, so it spreads easily, but then in an unlucky few does cause severe disease.
All right, well, thank you very much. This was really very, very exciting, and lots more to look forward to.
Yeah. Thank you. I mean,
that's a loaded statement, Abby.
Talking of lots more to look forward to, is anyone ever going to organize another meeting where we can actually get together properly rather than all this Zoom stuff?
Here.
Yeah, that's true. I think it's time.
I think it's time. I was slightly disappointed with this meeting that nobody, you know, I think we have to at least move to hybrid meetings. So people that can meet do meet, and anyone stuck in a country that can't join at least can join remotely. But we've got to start getting together, I think.
No, that's true. The American Academy of Neurology this year is all an in-person meeting.
Great.
Yeah.
Good. I must have missed my invitation to that one.
Well, we'll still send you an invitation.
You're always invited.
You're always...
That's the one where they invite you to come and talk and then they make you pay for it.
Yeah, that's right.
Anyway, great to see you guys. Yeah.
Take care.
Thanks so very much. Take care. Bye now. Bye.
Okay, well, I wanted to and thank all the speakers and for a terrific session and a really good discussion as well, and also all the organizers. And in particular, I wanted to thank Michael for the help that he's provided in helping organize it and managing today's session. Thanks very much, Michael. If you want to add a few words, you're welcome to.
Thank you for roping me in, Avi. It's always great to work with you on any project. And again, likewise, thank you to all the speakers and for a great discussion, and to the organizers as well.
Thanks.